All right. So today we have Shahin Nasr here. Shahin is a PI at the Martinez Center for Biomedical Imaging, and he is spearheading the Mesoscale vision research there. I think in the layer from my community, you're mostly known and celebrated and recognized for this ocular disparity in the color paper, which put you on the map for me at least, which I think is being cited a lot and has really started kind of a new research line across many labs, as we just talked about. So your title today is Mesoscale Cortical Plasticity in Humans Revealed by High-Resolution Functional MRI. Please, Shaheen.
Thank you so much. So Renzo and I agreed on a joke, but suddenly he said something that, I have to change my joke. My name is not Nasser. It's Nasr. And the reason that I correct people about that is that, we have a Dr. Nasser in US that is, he is infamous because of the abuse that he did with, US national gymnastic team. So I correct everyone who say that my name is Nasr. If it is hard to pronounce, just imagine that my name is Nas. Skip the R and just say Nas. It's totally fine with that. But anyway, so again, good morning, everyone. Happy Monday. It's a true pleasure to be with you. And as I was telling, Renzo, it's also a surprise. The reason for this surprise is that 2 or 3 years ago, when I published my paper with Brian Kennedy et al., so we contacted Renzo and we said that Renzo, we have done something great. And, this is a, mesoscale, organization of visual areas, blah, blah, blah. Have a look at it. You will love it. And in few minutes, we got the answer. I'm very sorry, it's 1 millimeter and we are not interested. That's it. So, and I think I got blocked after that. But when he invited me, said that, okay, so I'm not gonna say no to that. This is one once in a life, chance, and he probably will notice that, I'm the 1 millimeter guy, so he will skip me, after that. So thanks a lot for having me here. So as you see, the talk is, focused on cortical plasticity in human. And I imagine, you all have some idea what is, neuronal plasticity. So majority of time, when we are talking about neuronal plasticity, we are talking about brain changes in response to changes in the surrounding environment. This surrounding environment doesn't mean that 1 meter around you is about whatever is interacting with you. It can be through phone, it can be through TV. So it's not about a physical distance. It's actually about interacting with your sensory systems. And majority of time when we hear about a neuronal plasticity, we think about something like, oh, this is recovery from injury, learning new skills, something fantastic. We stick to the good stories. But this is Monday, people, and Monday is a bad day. So nobody wants to, start Monday. So I feel the same. It's gray. So I'm going to talk about developmental disorders. So there is nothing positive in this talk. It's all about disorders. And but hopefully through that, we will learn something about how brain functions. So the talk is focused on visual system and a developmental disorder that we call it amblyopia. Amblyopia is something that, is a disorder caused by unbalanced binocular vision in early stages of life. I think, through your life, you all have seen cutie pies like this, that they're walking on a street and they have one patch on their eyes. And when you ask the parents, they probably say that, he has a lazy eye. Lazy eye is another name for amblyopia. And majority of time through this patching, we try to encourage the person to use the weaker eye. Amblyopia is a phenomenon that affects almost 3% of children. And it's not a rare disease. It's actually a very common disease. So if there is one thing that you want to learn from this talk, is that please take your children to eye doctor very regularly, especially in early ages. Because if you don't do that and your child is one of the amblyopic cases, then for the rest of their life, they probably only use one of their eyes rather than using both eyes. They will have relatively poor to no depth perception or stereopsis, and they will struggle with a lot of visually guided tasks. So this is the main, take home message. Take your children to eye doctors. Amblyopia is actually one of those disorders that we have known about it for thousands of years. It's without any exaggeration. The first, in the, reference to amblyopia goes back to texts from Socrates. And they indicated that, some people without having any external, problem, they struggle with using one eye. So it's a very well-known phenomenon. But to this day, our knowledge of neuronal mechanism underlying amblyopia is based on animal studies. And this is mainly because the resolution of imaging studies or conventional imaging studies in human is not enough for studying amblyopia. Okay, so but what happens in amblyopia? We go to usual suspects. First thing first, it comes back to Odyssey. So for many of you that you're physicists and you start, wondering why everybody is focused on amblyopia, and they're focused on ODCs. One of the reasons is that physiologists that were focused on ODCs, they were also focused on those brain structures that are directly related to impairments. And ocular dominance columns are one of them. For those of you who are not familiar with ocular dominance column, which is a surprise if you are here, they are brain structures within first visual cortex that they respond preferentially to a stimulation of one eye over the other. You can see them like this as stripy patterns. Each of these stripes, they respond preferentially to one eye over the other. And there are columns. So it means that when you look at the cortical thickness, you can see them across all of the cortical layers. And one reason that they are super famous is that they, studying them brought Hubel and Wiesel Nobel Prize in 1981 because they were studying ocular dominant columns, their organization, their development, and also their neurophysiological properties. So if somebody wants to get the second, Nobel Prize, maybe they focus on that, hopefully in human this time. But anyway, so they are considered as building blocks of the primary visual cortex. But surprise, surprise, these are not the only mesoscale brain structures that are affected by amblyopia. And for those of you who are tired of hearing about ODCs, there are still other brain areas that are mesoscale, dying to get your attention, and they will benefit from high resolution fMRI even more than ODCs. The most famous one is LGN and LGN layers. We know that different LGN layers, they get input from either contralateral eye or ipsilateral eye, and they are more under influence of magnocellular pathway. Here you can see them with red, or parvocellular pathway. It's still one of the hardest tasks to localize them, to differentiate them from each other. This one is I most likely will be retired when somebody detect these. Maybe Mariana's students, they get that, hopefully, but look at the size of them. compared to the resolution, to the scale bar. And these are the main structures within the primary visual cortex that they respond to colors. And differentiating blobs from interblobs is very important for understanding V1 function, and we are still far, far from that. And even also you're interested in layer fMRI, that's the other challenge for you. And then this is almost my territory, V2 stripes, that you can see thin and thick stripes here. Detecting them is relatively easy in human, but still it relies on high resolution fMRI. You can see them. They were named thin and thick based on their size in monkeys, but in human, they are not really thin versus thick. We will see them, in few slides. But one interesting thing about this structure is that they are detectable even in newborn monkeys. So it means that, right after birth, this paper by itself is like one of the most interesting papers I have seen. They, immediately after birth, they started doing experiment under complete darkness. So the surgeon and experimenters were wearing night goggles during the whole procedure to make sure that there is no light at all. And they still detected ODCs in a monkey right after birth. And this is right 2 weeks after birth. You can see blobs, you can see stripes in both cases. So the question is, then where is development? So where is the neuroplasticity? Apparently from birth, we have all of these things. Development comes here. Majority of time when we are talking about the development, we are talking about connectivity between these stripes. So for example, if you remember from Mariana's talk, when we are talking about ODCs, ODCs are selectively connected to their alike ones. So it means that if an ODC responds to right eye, it's more connected to other ODCs that they respond to right eye rather than left eye. And also when it comes to V1 and V2 connectivity, it's not that they're randomly connected to each other. Blobs are more connected to thin stripes, while interblob areas that are more connected to pale and thick stripes. And the most important thing is these connections become more mature through time and with intact visual input. This is the point that amblyopia and many perceptual disorder happens. This is 2 weeks old. You can see that there is no selectivity in connection. When you connect one location in the cortex, almost all areas in the vicinity, they are show connections. But the more mature the case is, the connections become more and more selective. And this is the phenomenon that can be interrupted if the input from the eyes become atypical. When that atypicality happens, for example, in the sense of amblyopia, then we see abnormal brain function. This is what we see in a normal brain. The visual input is intact. And when we look at the selectivity of ODCs in V1, we see that we have a full range of ocular dominant selectivity from highly contralateral eye selective to ipsilateral selective and also binocular ones. But when the subject is strabismic, it means that one eye is deviated relative to the other eye. All of the neurons become highly ocular selective. And when it becomes anisometropic, which means that one eye is weaker than the other eye, majority of neurons or ocular dominance columns, they become selective for the fellow eye or intact eye, and they rarely respond to the amblyopic eye. All of that is the knowledge that we have based on animal models. But there are still tons of gaps in our knowledge. The main thing is, can we detect the same phenomenon in humans? If we detect this phenomenon, is it correlated with the severity of amblyopia when it is measured perceptually? And what is happening beyond V1, beyond ODCs, when it goes to higher level visual areas? So whenever I show this graph, I feel a little bit, embarrassed, because I said that, okay, so yes, I published this data in 2025, but the earlier version of the same data, it was published in 2016. And to this day, people keep coming back and said that, okay, so it is very hard to detect it. This is the organization of ocular dominance columns in the primary visual area in human subject. The main thing that we do for localizing these structure is we stimulate left eye, we stimulate right eye, we contrast the activity, and we see the map. But the thing that even the best of you do not pay attention to that is this. It takes 4 hours of a scan to detect this. It means 2 or 3 hours, 2 or 3 sessions of scanning to detect this. Majority of cases, people put the subject inside the scanner, they collect 2 or 3 runs, they come out and say that we see nothing. Yes, you need to keep scanning. So even when we use high resolution 7T scanners, it still needs tons of scans to detect these structures. And if you're a fan of Renzo and you want to go to higher resolutions, this is what you have to expect. 6 hours of scanning just of 0.8. And now the question is, just imagine if we use a Feinberg, approach, 0.4 millimeter. You need a subject that is willing to come back probably close to 20 or 30 scan sessions. That's the number that I guess. With 0.4 millimeter, the number goes to 20 scan sessions, unless you come up with a sort of, magic stuff to reach to that level. But this, yes, it's hard, but it also has a hidden message in that. To all of you who says that fMRI is not consistent and is not robust enough, look at this. This is one subject that has been scanned close to, if I'm not mistaken, 7 or 8 scan sessions. And this is the comparison of those scan sessions. You can see how close these scan sessions are. If fMRI was not reliable and robust, it was impossible to see these patterns. Even if you zoom in, then you see that the patterns are extremely similar to each other. And the other thing is, yes, going to a higher resolution is a pain, but still it's valuable because it can improve the level of differentiation of ODCs from each other. The higher you go, the better your findings and the more information you can get from your structures. So this is what you see in controls. After all of these things, it was something like, 2 or 3 weeks ago, I was in a conference, I presented this data, and people said that, are you sure this is not noise? And I said that, okay, I really don't know what you want from me, but they say that, oh, if you show functional connectivity of these ODCs, then I will accept it. And then, thank you, Mariana, you saved the day. And then that was the location that, that's the, condition that, Mariana's finding that she presented, I think 2 sessions ago, was important. You go, you check the resting state functional connectivity between these. You show that ODCs with alike ocular preference, they are more functionally connected to each other compared to unalike ones. And this connectivity is more apparent in middle cortical layers compared to superficial and the deep layers. This is something that is really hard to attribute to noise. And fortunately, When Mariana was presented that data was still under review. We were biting our nails to see, what we get from the reviewers. But now the paper is almost accepted, with one reviewer pulling our leg saying that share the data, and we keep saying that data is shared. Editors are not sharing them with you. So anyway, so what about amblyopia and developmental disorder? This is the data from a neurotypical, a control subject. When it comes to strabismic subjects, consistent with what I showed to you in the sense of single cell recording, you see that activity becomes stronger, patches becomes way larger, and you still see very strong ocular dominance activity. But the overall pattern become completely abnormal relative to what you see in neurotypical people. And then one very important phenomenon is that this activity is not limited to V1. You see very apparent extension of activity to higher level visual areas. If you are a reviewer and in the future you get someone submitting their paper about ODCs to you, and they mask activity outside V1, please ask them to remove the mask, because the main factor to clarify whether their mapping is correct or no is that in controls, these ODCs should be mainly limited to V1, and in abnormal cases, it always extends beyond the borders of V1. And the beauty is that when it comes to anisometropic individuals, you see a relatively uniform response to the amblyopic eye or dominant eye compared to non-dominant eye. And again, this activity is extended to higher level visual areas. So the second, first, point is that we can replicate what we know from studies in monkeys, and other, and also in cats or other non-human animals. But are these findings robust? For God's sake, it is robust. First session of scanning, second session of scanning in a control, in a strabismic, in an isometropic. And actually, this is a very valid point, because, when you're dealing with larger patches, these larger patches can be easily because of instability in scans, especially when you're talking about atypical individuals, because they have a problem with fixation. But you can see that actually in these individuals, the level of consistency is higher compared to what you see in controls. However, to some extent, this is because of larger patches compared to what we see in controls. And especially this is the case for anisometropic individuals. But still, it's a very, very consistent across the two sessions. And when it comes to the correlation between this ocular dominance activity and the level of amblyopia, which we usually measure as interocular visual acuity difference. For example, one eye is 20/20, the other is 20/100. The difference between these two, they call it depth of amblyopia or interocular visual acuity difference. And you can see that the more interocular visual acuity difference, the stronger OD response. And don't forget, all of these measurements happened after correcting the eyes. So it doesn't mean that for these individuals, there is a problem in their eyes. No, eyes are cured in these individuals. The problem is in the level of brain activity. So don't take it wrong. This is the measurement during best corrected condition. Okay? And if you are more interested in the details of these activities, you can, read this paper. Renzo, hit me.
Sorry, you lost me maybe on the last slide in this one. So the terminology, maybe you said it, maybe the one before, even like that in the terminology, you have the disease state of amblyopia, and then you have the strabismus and
Okay.
Anisometry.
Strabismus and anisometropia, that many people cannot pronounce it, are two subtypes of amblyopia. So strabismus is a type of amblyopia when one eye is deviated relative to the other eye. I showed it to you here.
Okay, oh gotcha. Oh, sorry, you
Okay.
did introduce it. Okay,
And then anisometropia is when one eye at birth is weaker than the other eye. But even when you remove that problem, the problem with the neuronal activity persists, probably for the rest of the person's life. So there
gotcha.
are both amblyopia, but two different subtypes of amblyopia.
Makes
Okay,
sense. So I do have a follow-up, and also Atana has
please.
her hands up. Is
You're the
now the time?
organizer, so you can ask. Okay, go on.
Okay, then maybe like everybody has a dominant eye, right? And usually you need to
Yes.
account for that in calibrating your task to some degree. But I guess that kind of at the end, not the entire visual cortex is dominated by one eye, right? You need to level it out, do you? And
So
then,
the difference between the two eyes in in a person with neurotypical vision is very, very small. So
so,
technically, when it comes to visual acuity, there is no real measurable difference between these two, eyes. We
gotcha.
call them dominant eye because we learn that whenever we need a very high precision, we have to close the other eye and use this eye. For example, when we want to shoot.
Right.
But when it comes to the visual acuity, there is no difference. And when it comes to brain activity, except for first visual area, no other visual area show preference for one eye over the other eye. No other cortical brain area show that preference. Did I
Gotcha.
answer your question?
Yes, you didn't do any calibration for one eye over the other on the left side of the graph.
Sorry, for the left side, what do you mean?
They're neurotypical.
Okay, so we usually, the calibration that we have goes to our devices, not to the visual stimuli. Technically, we use filters for making sure that when we present a stimulus to the targeted eye, the other eye is not seeing anything. And we want to make sure that the filters, they don't have any crosstalk. This is not about your visual system. This is about the filters that you use. So we just wanted to make sure that the apparatus that we are using is working fine. So for that, yes, we make sure we ask our subjects, do you see anything? With the other eye, they usually say that, no, I don't see anything. And I said, okay, so now let's switch it and then we go. But it's not about, calibrating the two eyes really.
Cool.
And yeah,
Thanks.
so you're welcome.
Atana, do you want to go ahead?
So my Hey Shahin.
Hi.
My first question is kind of related to just what Renzo asked.
Mhm.
Your paradigm, if I'm not mistaken, is right eye versus left eye.
Okay.
So you don't have a baseline between right eye, right eye, and left eye,
I have
right?
baseline at the beginning and at the end of
And at
experiment.
the end, again, yes,
Yes.
but not between the condition
No,
itself.
no,
So,
no.
well, we have used two different paradigms for our experiments. I just wanted to ask you if you have looked at the responses of right only and left only. And if
Yes.
you do so, how much of overlap would you expect to see between these responses, if there is any?
Okay. I will be more than happy to discuss it with you, but at the end of the talk.
Okay.
So,
Okay.
because it's a very valid question, the answer is you cannot differentiate the response of left eye and right eye when you compare it relative to baseline. So the pattern, when you look at it relative to baseline, is they are identical, almost identical, unless you go to local, very small regions and you play with your threshold, then you will see that. But when it comes to the whole V1, if you play with the thresholding of whole V1, it's almost impossible to differentiate the two relative to each other. But I will be more than happy to discuss it further at the end of the talk.
Okay,
Okay,
thank you. So just one more question regarding
sure,
the correlations that you just showed between
sure.
the sessions.
Okay,
So
let me go there.
correlation,
Okay, please.
yeah. By session here, you mean the long 8 hour session
Yes.
or
No, no, no.
just
By that one means that, one session of 2 hours versus another session of 2 hours.
okay. And 2 hours is
Two
one
different days.
single imaging session.
Yes.
So, okay, thank you.
So we measured, this is the ODC map from one subject at the end of one session. This is a ODC map from the same subject's second day. And we are correlating these two maps relative to each other. And this is the correlation that we get. So, okay. And you may say that, okay, so thanks for doing that. What's the value of, doing this when it comes to clinical studies? This is actually one of those things that, makes, clinicians jump up and down or makes them very sad, actually. This is the scan from someone who, based on the clinical factors, they have recovered from amblyopia. Recovery from amblyopia when it comes to clinic means that the acuity of the two eyes become comparable relative to each other. In clinical measurement, we typically call this 0 and 0. So here, the difference between the two eye is almost non-existent. The person used to be amblyopia, and now at the end of the treatment, the acuity of the two eye is completely comparable relative to each other. The person still has some diplopia or dual vision, and their dominant eye is the right eye. They still have a little bit of misalignment, but is not huge. And as you can see here, the pattern of ocular dominance activity is still completely abnormal. This is the value of functional MRI in human, that it shows that in many cases, when we call someone recovered from a disorder, and the measure for that recovery is purely based on behavior, it doesn't necessarily guarantee that your brain organization is typical now. It just means that your subject has found a way to improve their performance through training. It doesn't mean that they are using the same method as your controls. And because of that, a lot of these individuals, after a few years, they go back to amblyopia. They recover from amblyopia, they relapse to amblyopia. They go to amblyopia, they come back. They go, they come back. And because of that, they never fully recover. So full recovery from amblyopia is a very, very rare case. So don't forget that. And this is why we need more studies like that in clinic to make sure that the person is completely recovered. And this is the study that Renzo very, very politely kicked out. So we tried to technically open the door for going beyond ODCs and say that, okay, ODCs are fantastic, but what happens beyond ODCs? And this is very, very important because even in monkey studies, nobody has gone to these regions and see what is happening in amblyopia. The study at the time was so groundbreaking. I just want to, give a harder time to Renzo. Was so groundbreaking because it was a COVID time. And during COVID time, we did 150 scan sessions to collect this data. And then we showed the data that there is no equivalent of that even in healthy cases in animal models. Anyway, the point is when it goes to higher level areas that we usually call them extra straight visual cortex, V2, V3, V4, you see a stripy patterns that they are either motion selective, stereo selective, or 3D versus 2D, or color selective. that they respond more to color versus black and white. And the beauty of these maps is that you may say that, okay, so they're just a bunch of noisy pattern. It's not noise, because when you put them on top of each other, they become completely interdigitated, and they just make the map more complete. So you see that the blue, green, and red, they are completing each other, and the level of overlap between them is not really high. And when it comes to the response property, the response properties significantly differ between them. Motion and disparity selective ones, they show more selectivity for low frequency stimuli. This is, one of those things that, Renzo and I talked about them multiple times, that how we can actually judge whether we have differentiate clusters from each other perfectly or no. It goes back to the function of those clusters. If we have a good localization, then the function of these clusters, they are also differentiable from each other. When you're dealing with the smoothing or other artifacts, these functions, they are less discriminable from each other. And here you can see that they're clearly discriminable from each other. And then when it comes to amblyopic individuals, that it took us technically 3 years, and now we have a good preprint, almost ready to make it public, in controls, strabismic and anisometropic. I told you that anisometrops, they struggle with depth perception or disparity selectivity. And then you can see that in these individuals, you still see motion and color selective areas, and they are still completely interdigitated relative to each other. But when it comes to disparity selectivity, disparity selective activity in these individuals almost gone. But the overall interdigitation, there's still there. The level of drop in the stereoselective activity, which we measure as 3D stronger than 2D, is correlated again with the depth of amblyopia. So the worse the perception, the smaller the level of activity. And you can see it in both in V2, in V3, and in V4. And this is one of those things that even in monkey studies, it was still completely unknown. When it comes to the level of disparity selectivity, you can see that there is a very apparent drop in the level of disparity selectivity or stereoselectivity in controls relative to amblyopes. But the beauty is that the level of motion and color selectivity is going up. There are more sites that they show motion and color selectivity. This is a new phenomenon that we try to introduce as a developmental rivalry. So it means that when you are not using one visual feature in the brain, rather than letting those neurons die, the brain starts using it for encoding other features. This is something that in the past you probably heard about it in the level of areas. Like when somebody is blind, visual areas, they start responding to other features. But this is by itself one of the first examples of developmental rivalry in mesoscale levels. And it shows that even in the level of columns, there is such a rivalry. And the beauty of that is there is no equivalent of that in monkeys. This is data that is first presented in humans. And okay, I'm almost done. So hopefully, first I have convinced you that I'm one of you guys. We do some high resolution fMRI. And based on using this technique, even with 1 millimeter, you can still visualize neuronal plasticity, which is very valuable for both understanding visual perception in controls and also in individuals with neurodevelopmental disorder. And based on that, you can link perception to mesoscale functional organization of the brain. But even if you do 1 millimeter, scans in future, you can still do a lot of things. Even if Renzo doesn't advertise you guys, we still don't know if there is a direct link between the connectivity and selectivity. And this is one of those projects that hopefully in a year, I will be desperately looking for interested postdocs to come and start doing that. So can we predict selectivity based on connectivity? Can we predict atypical brain activity based on atypical brain neuronal connectivity? And then what is happening in adult? In majority of cases, when we are talking about developmental disorder, we are talking about children. What happens when it comes to adults? Somebody who lost one eye when they are adult. Do we still deal with neuronal plasticity? There are tons of visual disorders that they fall under this category. We can also talk about, because Birkin is here, we can also talk about disorders and comparing a structural connectivity relative to functional connectivity. And that's why paradigms like high resolution DWI become super important for us, because we need to compare them relative to what we have in high resolution resting state fMRI. And now, just thanks everyone for listening to me. I'm very talkative. I know that. Thanks to Renzo for tolerating me and my jokes, and also my wonderful colleagues, Peter Becks, Eric Geyer, David Hunter, especially Brian. If you get any application from Brian, please accept him. He deserves a lot of support. And Nikita and also Tony. Mariana is here. She's looking for a postdoc, position. And, if you get her, I think you will struggle with her attitude, but, she's a wonderful person. And then also Jan and my supervisor for the rest of my life, Roger Tutel. And thanks for to my sponsors. Okay, hit me.
All right, thanks so much, Shahin. I do feel I should explain myself. I looked it up now. It was in December 2022, and you contacted me on Twitter and you
Yeah,
asked to advertise it. And
yeah.
there has been, like, I have this claim of completeness of all the human layer dependent fMRI studies, and it was a process to get there, and there was a lot of political kind of compromise, and then kind of I think there are three criteria that had to be fulfilled to be kind of included or excluded in this list. Otherwise, anyway, people might have complained. And yes, it's absolutely it's an arbitrary choice of the 1 millimeter thing. But that's why I think I did say it's a cool study and I like your figure, but I didn't include it in the
You
list.
definitely, and Renzo, first thing first, thank you for letting me making all of these jokes. You took one for the team, so
Yeah.
that's it. And it was just an icebreaker. Otherwise, thanks a lot for everything that you have done for the field.
Come on, yeah, sure. Next time we just acquire it with 0.01 millimeter smaller boxes, shall we?
Yeah.
And then it's
Yeah, no,
0.99.
I know. I was thinking about that. I could say that is, point 9999. So, who could, say no? Okay,
Are there
so
specific
Atena,
questions?
so did I answer your question, or do you still want to follow up on that?
I
I
do
do
have.
not have those. It's my apology, Renzo, but I do not have those slides handy, but I do have them somewhere, so I can show you the response to one eye, and we can discuss it further. But I expected actually one more question for you, considering that you work with, Robbie, and the fact that is there also a difference if we had a gap between conditions? If we had, blank presentation between condition, do you think that, there will be a difference between, maps? There could be, because Ravi
Well,
has a, couple of, hypotheses on that, but I never tested that. So please.
Well, the maps that we have created so far hasn't gotten any attention. The reviewers are like, what you have measured are basically noise and artifact. They are
Oh,
not an actual ocular dominance columns map. So
okay.
I have tried two different paradigms,
Okay.
right eye, right eye, left eye with the baseline at the beginning and at the end. And we also tried a paradigm with the baseline interleaved between each eye condition.
Mhm.
So
What was the stimulus?
uh, the stimulus is a checkerboard, flash and checkerboard, 8 hertz.
Okay. Did you try it with random no random dots?
No, but I've seen that in your paper and also Daniel's paper
Yeah.
that you guys have used that. We have used checkerboard pattern,
So
but
so so that
unfortunately, no luck.
no, I know, but I imagine that I think that Feng and others from China. Sorry, my apology. I don't remember. Where is the the exact institute? Renzo, do you know?
Peng Chong, yeah.
Yeah.
Uh,
So where
he's
is the
in Beijing, yes.
Okay, so it's Beijing. I think they used also, flashing checkerboard. I'm not sure about the contrast of, those flashing checkerboard that they use, but generally speaking, I think, random dots are better as stimuli. And the only reason that I say that is, my assumption is that there is a good chance that flashing in checkerboards take you to saturation level. And when you have a saturation level, differentiating left, right, and right eye from each other becomes harder. However, I never had the budget and a person, a student that is actually very interested in this topic to go and check why people struggle. Because all of these are very valid questions. We have to check all of the paradigms and say that why moving random dots, they generate this. We have done that. Vase group, and Daniel has done that. Feinberg group, they never acknowledged that they got their stimuli from us, but they used the same stimuli and they they did that and they showed, ODC. So many people successfully used these stimuli and did that, but I never had the manpower to test it. Why?
Yeah,
And I have a feeling that if we change the stimuli, we may see a minor difference. But I have very limited data on that, and I need people who are interested in collaboration. And anyone who is interested in this topic, just drop me an email.
I will, sure. Yeah. And also our scan times was not that long. So it was 1 hour for laser scan and 2 hours, almost 2 hours for the bold scan. Um, and we did 2 sessions per participant. So that might also be one of the reason that it's very noisy and
Happy to
not,
chat about that.
yeah,
Happy to chat
yeah.
about that. Yeah, sure. Okay.
If nobody else is coming forward, I have a question.
Oh,
Maybe kind of more brainstorming or kind of,
please.
I find your maps very inspiring and the kind of the stripiness. I'm always surprised how few stripes there are, right? That kind of maybe there are a dozen or so. Um, across the entire visual field. And then kind of especially as people are interested in laminar fMRI with respect to retinotopy, I was wondering what you think about the ultimate resolution of retinotopy. So we would expect that within stripe, kind of the polar angle or kind of eccentricity would kind of then go as a sawtooth pattern. Are these stripes basically the spatial resolution limit of eccentricity in retinotopy?
Okay, so first, please note that this is just the central 10 degree. This is not the
Right.
whole V1. So when it comes to, more peripheral regions that, the wonderful goggle that we have in Bay 2 and nobody used that. If one day we start scanning again, we have to be able to see blind spot. And then when we go even further, you're completely correct. These, stripes become very, very, expected to be much smaller and less stripy pattern when you go closer to monocular crescent. So what is the resolution that we want to, we need to do that? Probably it's not achievable right now, but my guess is, we probably need to go closer to, 0.1 millimeter to be able to, clearly detect those stripes. And to be very clear about that, when it comes to these maps, when it comes to anisometropia, I don't think that these maps are 100% correct. I think these maps, if we go to higher resolution, we still see some dots that are representing the other eye. It's just a matter of limited resolution that we see them as a uniform yellow. So this is still far from the resolution that is optimum for amblyopia studies and also ODC studies. But it can answer a lot of questions. Did I answer your question,
Oh, yes,
Renzo?
absolutely.
Okay.
So these stripy patterns, kind of the shape of it, and it's basically, I guess, projected onto the surface. And in gradient echo bold, we kind of have these large trailing veins that are annoying. And you have this very elegant way of kind of having both task conditions perfectly orthogonal, right? They have the same strength to kind of
Almost,
distinguish them.
yes.
So,
Yes.
but based on the shape, kind of if we would have a large training vein that I guess pulls more from some of the ODCs from the left eye or some of the ODCs on the right side, that vein will also always be kind of somewhat survive that differentiation. So to what degree are you sure that these kind of stripiness are
Yeah.
really the neural column stripiness,
Okay.
or could there be some stripiness of the veins also elongating them, making them more stripy-like, because the veins are elongated,
Yeah.
right?
So there is a reason that Renzo and I are good friends because, sometimes, I go to him and I say that, Renzo, this is a concern in the field. And then Renzo comes to my own talk and, give back, the same concern so that, yeah, this is the question. So Farouk, if you one day, you listen to me, this is why we need you to do vascularization mapping. And then we can answer this question. So generally speaking, there are many studies in monkeys based on optical imaging that is more or less similar to this, fMRI, with the difference that they can detect the veins there. And except for couple of or cherry picked, the images, nobody has ever found a vein that follows ODCs. And the other factor is that if these were veins that they were following ODCs, then actually you should not see them for strabismic and anisometropic, this pattern. Then even in a strabismic and anisometropic pattern, you had to see exactly the same pattern as in controls. Because veins are veins. Veins are not going to change based on amblyopia. Nobody at least has shown such a thing that, veins, they become, instead of feeding, for example, the right eye, they start feeding the left eye. No, the structure remains the same. So in that sense, you expect to see the same pattern as controls. But still, a lot of these answers are hand-waving answers. The best answer is let's go there, let's map the vascularization and then compare it relative to that. The same argument is also correct about V2 stripes, and actually it's more possible there. We can go there and we can check it. So I never claimed that we have reached the end of science. So no, there are still tons of questions left.
Yeah, great. That's what I wanted to hear. Yeah, let's
Oh,
look at
yes.
it. Yeah.
Yeah, sure.
Maybe last question. So you made a point of kind of how much scanning we need.
Yes.
And I guess it's a really different effect size if you have kind of a flickering checkerboard versus nothing compared to the effect size of left eye versus right eye at this resolution. But still on the right part of your slide here, you see a beta value of 2. Is that percent? Because 2% would
Oh,
be
no, no, no,
quite,
no. This
no.
is yes, this is beta value. Yes.
Like 2% is not that bad, right? I
No, it's
realized that
not.
only some of the voxels really
Yeah,
exceed
some
there.
of the voxels reach there. Yes.
Um,
Yes.
So I guess in parts of the primary visual cortex, you might even see stuff kind of in shorter scan times, I guess.
Yeah, but yes, probably. But the problem is, you look at it from the methodological, point of view. I look at it from the physiological point of view. If I show a pattern just in a part of a V1, then I cannot defend it as ODC. Because people will say that ODC is expected to be, detected in the whole stimulated part of V1, not just in a part of that. But for you, it's more acceptable because you say that, okay, so I see that the reason that I see it here but not there is because of vascularization or other artifact that they exist. Okay, but it's again, it's because of our audience. My audience are neurophysiologists, clinicians, that they just look at technique part as like a sort of, knife in the hand. Good, you have a knife in your hand, but I'm not really interested in your knife. I'm interested in the outcome of your study.
Makes sense, yeah. We are at time and people are dropping off here. So I want to thank you, Shahin, for presenting this. Very entertaining and engaging. And
Very
there will
welcome.
be no layer seminar in 2 weeks because there will be the ultra high field workshop at CMR with the layer fMRI hands-on pre-workshop. So we will meet again in 4 weeks talking about isomer abstracts. Thanks a lot.
Bye.